Students, in the last video we have seen the process of fertilization, and after that the formation of zygote in the region of the ambulary ischemic region we have seen in detail. Now we'll see embryonic development, and in that embryonic development, in that part, it will include the process of cleavage, first cleavage, second, third. Then after that we'll see morula formation, blastula formation or blastocyst formation, and after that we'll see implantation. In that part we will see also the formation of all these three germinal layers, and that process is known as gastrulation. So let us start. First of all, the zygote, how it will undergo the first cleavage. Already we can understand that whenever this zygote has been formed, that zygote will carry twice n chromosome, half n from the paternal side, that is from the spermatozoa, and another one, that is n, that will come from the egg itself, that is from the female. So after that it will have, this is the zygote, it will have twice n number of chromosome, and this zygote will be surrounded by the layers, the egg itself. This is the primary layer, which is known as the vitelline membrane, or we can also call it as the same plasma membrane. Around that there is a non-cellular layer present, and this non-cellular layer is the zona pellucida. The very important function of zona pellucida is, first of all, it will actually check polyspermy. That means, so that more than suppose one sperm, if come to the region, that will be not allowed. Only one sperm is allowed to fertilize the egg. So it will check polyspermy, it will block polyspermy, and around that it will have the corona radiata layers. So here the corona radiata cells that will be still present. Only in one region the corona radiata cells are little bit less, or from that region it is dispersed. Why? Because the sperm lies those corona radiata cells, and that's why actually in those regions the corona radiata cells are a little less. So that region where the corona radiata cells are a little bit less, that means from that region the spermatozoa have fertilized the egg, that region, this is known as animal pole, and exactly opposite region, this is known as the vegetal pole. From the animal pole region itself, the second polar body, that will be also removed outside. During the second meiosis, that second polar body was formed, and that second polar body will be also removed from that animal pole region, and both the nucleus from the sperm and the ovum will merge and undergo the process of formation of a diploid cell. So this is the zygote. Now after that we'll see the cleavage. So how this cleavage formation, first cleavage will occur. See, after 24 hours the first cleavage will occur. First 24 hours after fertilization. Suppose fertilization had taken on that time period, exactly after 24 hours, it can also plus minus 1 hour later or 1 hour before. So within that 24 hours, the first cleavage will occur, and that type of cleavage will occur from animal pole, vegetal pole region. So from the animal pole and vegetal pole region, first of all there is a formation of cleavage furrow will occur, and whenever this cleavage furrow will occur, gradually this will form two cells. So see, first of all cleavage furrow is formed, and after that it will completely divide the cell. Now here the zygote, which contain twice n chromosome, now each of them they will also have twice n chromosome. Now the same layers, that is zona pellucida and corona radiata cells, will be still intact. So here the zona pellucida and corona radiata cells will be still present. So they will have numbers of function, we'll see in detail later. So first of all it will have the corona radiata and zona pellucida layer. So here we have to see what type of cleavage is that. First of all, this cleavage will occur. First, we have to see the type of cleavage. First of all, first cleavage occur after 20 hours of fertilization. After 24 hours of fertilization, the first cleavage will be formed. That type of cleavage that will form two cells. These two cells, each of the cells, they will be known as blastomeres. They will be known as the blastomeres. Now, one thing you can see here, the zygote, and after that it formed the two cell. After first cleavage, both of them, they will have same size. Just here what happened, division is going on, but the cytoplasm content is remains same. Only the nuclear division is going on. So that's why in every cleavage, every after the cleavages, what happened, the cytoplasmic content, they will remain same. So that's why the cell size will start to decrease. So here what happens, after 24 hours of fertilization, first cleavage has taken place. Then it will form two equal sized blastomeres. Two equal sized blastomere will have less cytoplasm content as compared to the zygote. Next important thing you have to know about the cleavage, that this type of cleavage is indeterminate. Indeterminate means what? Indeterminate means that each of the cells, every after cleavage the cells will go on increasing. They will be not fated with any type of future formation of cells. That means each of the cells, they can form any type of tissue. That means in the division, earlier it is not fated that they will form the nervous tissue, they will form the ectoderm. It will not happen. Any cell can form any type of tissue, but this is not happening in case of some other organisms, lower grade of vertebrates. That in case of lower grade of vertebrates, it is already determined that which cell will form what type of tissue. Suppose some of the cell will form the epithelial tissue, some of the cell will form the nervous system, some of the cell will form the muscle cell. That will be already predetermined. But this is not happening in case of human. In case of human, it is indeterminate. That means any of the cell can form any of the types of tissues. Next type of another very much important type, that this cleavage is holoblastic. Holoblastic means what? It is the first cleavage which is later will go on dividing, and every after the cleavages, what happened, the cell number is increasing. Here you can see that the cells are dividing to form two completely equal sized cells. So here what is happening, the zygote form two cells. Each of the cells that will be from the meridional axis, from the animal pole, vegetal pole axis, it is dividing and form two cells. And this completely divided two cells, that means in between the two cells there is no common passage or there is no gap junction, nothing is present. Completely the cell is divided into two cells. But what is happening in case of some other organisms? See, holoblastic cleavage is occurring in the type of egg which have very less yolk content, which do not have yolk content. You can see in case of human, the egg yolk content is very less. So that type of egg is known as microlacetal egg. So in case of microlacetal egg, what happened? The cell completely divides to form two cells. But this is not happening in case of other vertebrates like the reptile and the ape. In reptiles and ape, what happened? They have much amount of yolk present. So that type of egg is known as macrolacetal egg. So in case of macrolacetal egg, what happen wherever the yolk is present, suppose yolk is present here, so where this yolk is present, in that region cell division do not occur, which is not happening in case of microlacetal egg or in case of human egg. In case of human egg, as the yolk content is very much less, so that's why the two equal size cell is being formed and there is no region for the presence of yolk, or in that region the cells is not dividing. It's not like that. It is completely forming two equal size cell. So one thing you should know about the indeterminate type of cleavage, that is very much important characteristics of the deuterostomes. Deuterostomes, what are the deuterostomes? You should already understood in the first year. In the first year, from the echinodermata up to all the vertebrates, up to mammalia, all of us, we are deuterostomic animal. Deuterostomic animal means that from the blastophore, that means in the embryonic development, first of all anus has been formed, and after that the mouth will be formed, or the anus, first of all anus will be formed, and after that mouth will be formed. But in case of prostomic animal mean right from the porifera up to the echinodermata, that means up to mollusca, this type of organisms are prostomic. That means they form first of all mouth and then they will form the anus. So those group of organism, the cleavage is determinate. That means already it is predetermined that which cell is going to form what type of tissue. Suppose some group of cell that is already determined to form, suppose epithelial tissue, some of them they are already determined to form the muscular tissue. Suppose some of the tissues are already determined in the cleavage stage itself, they will form the nervous tissue. So in case of deuterostomic animal, this is not happening, that each of the cell have the capability to form any types of tissue, and that type of cleavage is indeterminate. And second one, that is holoblastic cleavage. That means each cleavage will form the two equal size cell. That means in this stage what happens, each of them they will form two cells. So here this is a characteristics of the microlacetal egg. That means those eggs which have very less yolk content, they will form two equal size cell. See, first of all, after first cleavage, the zygote divide to form two equal size blastomeres. This is after first cleavage. After second cleavage, just see what happened. Around 30 hours later of fertilization, we can see another cleavage, and that cleavage already we have second two cells. After that, two cell stage is there. So after second cleavage, first of all at right angle to the first cleavage, earlier the cleavage was occurring from this to this, from animal to the vegetal pole. Now exactly at right angle to the first cleavage, we will have the second cleavage. And whenever second cleavage is going on, then we will see a transient 3-cell stage. That means in this stage, for some time you can have a cell of mass that is of only 3 blastomeres. So this is the transient 3-cell stage. So after that stage, we will have the complete second cleavage, and after that it will form the 4-cell stage. So after this, you can have a little half cleavage. That means second cleavage is not completely done. We have a transient 3-cell stage, and later of this transient 3-cell stage, we will have the complete second cleavage, and after second cleavage it form the 4-cell stage. So here this cleavage, see, after the second cleavage we will have a 4-cell stage. in this 4 cell stage, you can see like here we have seen that the zygote had the highest amount of cytoplasm. Gradually the cleavage is going on and the cytoplasm content as it is remained same, so each cell is getting less amount of cytoplasm. So gradually the cell sizes start to decrease. So after the second cleavage we have the 4 cell stage, and this 4 cell stage, what happened to second cleavage? This is meridional, and second thing that will occur, that cleavage is occur at right angle to the first cleavage. So first cleavage was occurring from animal pole vegetal pole axis, and the second cleavage is occurring at right angle, that means this way. So in this stage we will have the 4 cell stage. Now here you can see already this zona pellucida and the corona radiata cells are still intact. So here also you will get the same zona pellucida, and around the zona, just outside the zona pellucida, you will still have the cells of corona radiata. So corona radiata cells are still present like this. Till now the corona radiata cells are not separated, it is still present. After that you can see that third cleavage will occur. So after that, what happened after third cleavage? So you can see after third cleavage you will have a stage of 8 blastomere. So after around 60 hours later of fertilization, so after 60 hours of fertilization we will have the next stage, that means third cleavage will occur, and as a result of third cleavage it will form 8 cell stage. So right after that the type of cleavage is not differentiated, that means we are not able to understand what type of cleavage will be that. So that's why we can see here after third cleavage we will have the 8 cell stage. So after 8 cleavage, after third cleavage, it will form a 8 cell stage. So gradually you will have the structure. See, this is known as the morula. See, after third cleavage we will have 8 cell stage, and after fourth cleavage, we will have a 16 cell stage. So that 16 cell stage, what we can call it? We can call it as the morula, and this morula is look like the mulberry cocoon, so that's why it is known as the morula, and this is the 16 cell stage. So here suppose we have 16 cells, so this 16 cell stage we can call it as the morula. After fourth cleavage this morula has been formed, and whenever this morula has been formed, gradually from the outermost region the corona radiata cells start to disappear, but still zona pellucida will remain. So up to third cleavage you can see the corona radiata cells, but gradually this corona radiata cells start to disappear. So this way we will have the 16 cell stage. Here a very important reaction is happening, that is compaction. So you can see what is actually compaction. Compaction means in this stage what happened, the cells which are smaller in size they will go to the periphery, and the cells which are of large size that will remain inside. So this rearrangement of the cells, what we can call it, this is the reaction, compaction reaction we can call. So in this stage actually this is the same 16 cell stage itself, but the cells which are very small, have less cytoplasmic content, they will go to the periphery, and which are larger in size, those will remain inside. So this is a stage, this is the compaction. Here this stage is same 16 cell stage, but in that 16 cell stage you will get a region, a specific time when the smaller cell will go to the periphery and the larger cell that will remain inside. So this reaction is actually compaction reaction. So remember this after, and yeah, this morula itself will undergo compaction reaction. So you can see first cleavage, second, third and fourth cleavage. Right after each cleavage, the cell number start to increase, cytoplasm content start to decrease, and after third cleavage, the differentiation, the type of cleavage we cannot able to understand. So here the region where those whole reaction is going on. See, this is suppose the fallopian tube. In the fallopian tube, in the ampullary ischemic region, we had the formation of the zygote. So suppose here the zygote was formed. Gradually, the second cleavage had occurred, so it will remain a little bit towards the fundus region. So towards the fundus region it is start to move. So after that we will have the transient 3 cell stage, then after that 4 cell stage after second cleavage. So gradually what happened, after third cleavage we will have 8 cell stage, then after that we will have the morula stage where it is 16 cell stage. It is actually moving towards the region of the uterus. So whenever it is in the morula stage, what happens? What happened is coming towards the region of the implantation. That means towards the lining of the uterus, we have the 3 membranes, outer perimetrium, middle myometrium was there, and this endometrium was present. So towards the region of the endometrium, it starts to come, and after that it is going to have a reaction, and that reaction is known as the decidual reaction. So this morula will start to come towards this endometrial lining, and after that it will have a reaction, decidual cell reaction. So let us see what is that decidual cell reaction. So let us see what is that decidual cell reaction. First of all, let me draw that endometrium lining. First of all, this outermost one is the perimetrium. Just middle one is the myometrium where the blood vessels are present. And here, this is the myometrium suppose. And the innermost one, this innermost lining is the perimetrium. This will be the endometrium. So this is the innermost lining, this is endometrium. So here this endometrium will have a reaction with the morula, and after that will have a reaction. That reaction is called as decidual cell reaction. So let's just see what is that decidual cell reaction is. First of all, already this morula is coming towards the region of implantation. So suppose this is the 16 cell stage, and this 16 cell stage, this is morula which is still going to have this outermost zona pellucida layer. So zona pellucida layer will be still intact till now. So it is coming towards the region of the suppose implantation. So whenever it is moving, suppose it is moving towards this region. So whenever this morula is coming towards the endometrium, then locally a edema will be formed. Edema means a local swelling in the endometrium will occur. So in the endometrium region, a little swelling will occur. The cells will start to swell. It is going to have amount of fluid present. So fluid is still there. One very important reaction thing is happening, that the fibroblast cell, suppose this is a fibroblast cell, so that fibroblast cell, they will convert into a glycogen cell. That means this fibroblast will have glycogen inside it. So that cell will start to enlarge also, and here in this time actually those cells, those 16 cell stages there. So just see what is happening here. So in this time period, whenever this edema is happening, edema is occurring in the endometrium, this endometrium start to secrete a fluid, and that fluid will start to be absorbed by this morula. And whenever those liquid, see, a liquid will be absorbed, that is released from endometrium, that will be absorbed by this morula. So this morula, whenever absorb this fluid, this nutritive fluid actually, then the size of the morula will start to increase. See, in the same time, what happened? These cells will start to increase in size. So this fluid, whenever it has been absorbed, then there is a reaction is happening. See, whenever this fluid will start to increase, that fluid will start to present here somewhere in the middle. So what is happening? Those liquid, that liquid will start to present here, and in that period of time, that liquid will be present inside it. And whenever that liquid is present inside that morula, a cavity is formed. And that cavity has been formed. That cavity is known as blastocyst. And some of the cells, there already, I told already that some cells which are going to the region of the periphery, so those cells, what we can call it, they are known as trophoblastic cell layer. So just see here, those cells which are going to the region of the periphery, they are known as trophoblastic cell layer, or simply trophoblast. And some of the cell which is present inside it, see, those cells which are present inside, suppose these cells are present inside, they are known as the inner cell mass. So these are the rearrangement of the cells and the fluid occurs. So just see here, the cleavage will be still going on. So what happened, from 16 cell stage, it will form 32 cell stage. So in that 30, and gradually after that 5th cleavage, and this way the cleavage will go on. So from this morula, what happened here, this endometrium, they will start to, see, this endometrium start to form a local edema. Local edema means only swelling occurs only in the region where the morula is coming. So what happens, some of the cells that will start to have more liquid inside it, so that liquid will actually be absorbed by this morula. Some of the cells already go to the periphery, which are smaller in size. They are known as the trophoblastic cell layer, or they are also known as trophoectoderm. Some of the cell which is present inside, which are bigger in size, they are known as the inner cell mass. And that liquid released by the endometrium, and an amount of enzyme that is released from the trophoblast, that will be present inside the same structure, and that is present inside a cavity that is known as blastocyst. And whenever we see this type of thing, that means outer trophoblast, inner cell mass and blastocyst, which is filled with the liquid released by the endometrium, that is known as blastocyst. Now this blastocyst, this is the morula and this is the blastocyst. So this blastocyst start to engulfed by the endometrium. See what is happening. This endometrium will completely engulf this blastocyst, and gradually the zona pellucida layer will start to degenerate. What is the main function of zona pellucida? Zona pellucida help in implantation and prevent the blastocyst to get implanted in any another region. Suppose here the blastocyst is formed here. This will be not allowed. This blastocyst is not allowed to get implanted somewhere in the fallopian tube. So this zona pellucida will prevent it. So what happened? Maybe this is possible sometime. This implantation process can occur in the fallopian tube. So that type of implantation, if suppose implantation occur in the fallopian tube, that is known as ectopic pregnancy. This is known as ectopic pregnancy, and that ectopic pregnancy is dangerous for both the mother and the embryo, both for the baby also. So here, suppose in normal case we have implantation, and after that implantation, it is believed, it is said, and in some books you will get different times also, but around 7 days later this implantation had occurred. And is this type of implantation, when the blastocyst is completely engulfed by this endometrium, that type of implantation is known as interstitial. So interstitial implantation means when the whole blastocyst is completely engulfed by the endometrium, that is known as interstitial implantation. Now just see what is happening gradually in this interstitial implantation. Already understood what is interstitial implantation, when endometrium completely engulfs the blastocyst, or this blastocyst is completely submerged, it will be completely buried in the endometrium. That type of implantation is known as interstitial implantation. Second thing, a very much important thing is happening here, that this reaction is happening, that is known as decidual cell reaction. So what is decidual cell reaction? In the decidual cell reaction, what happened? Around this 7 days later after fertilization, what happened? The morula is coming towards the endometrium where the implantation had to occur. So suppose in this region it is coming, the endometrium locally develop a swelling or edema. Some of the fluid, nutritive fluid, it will be released by the endometrium, and it will be taken out by the blastocyst. So that's why, to have this liquid inside the blastocyst, they develop a cavity where the liquid will be present, that is known as blastocyst, where the liquid of the endometrium is present. Next one, some of the bigger cell that is present inside, that is known as inner cell mass, and some of the cell which is present on the periphery, that is known as trophoblast. So in that period of time, what happened? Gradually the zona pellucida. So see what happened, the zona pellucida start to fall, they start to disappear. So zona pellucida performed its function. What was the function? It completely helped this blastocyst to get implanted somewhere other region. And suppose somehow zona pellucida cannot perform its function properly, suppose implantation had taken place in the fallopian tube, that will be known as the ectopic pregnancy. Suppose now in this normal case, it is implanted in the normal region in the uterus itself, then this type of implantation properly had taken place in the exact location, and that's why gradually the zona pellucida will start to disappear. Now just see what happened in that very same time. What happened? Sometimes this is possible that whenever this liquid start to increase inside the blastocyst, pressure start to tremendously increase. Whenever this zona pellucida start to degenerate from that region, maybe it is possible that some cells that can be popped outside, and that cells, that mass of cells somewhere get implanted in the another region of the uterus, or exactly in the same region. So that may be the cause of twins, and that type of twin will be monozygotic twin. As already I had told you that this type of cleavage is This type of cleavage is indifferential. That means it is not predetermined that which is going to form which cell. So any of the cell can perform its function properly, and it can form the whole baby or the whole embryo from any of the cells. So that's why here may be a region of monozygotic twin, where due to the increased liquid inside the blastocell and zona pellucida start to disappear due to high pressure. Some of the cells can disperse from that blastocyst and they get implanted somewhere in another region. So that type of implantation that can result in the formation of monozygotic twin or identical twin. So both will have the same sex also. If both suppose the male, then both of them will be male, or same female also, they will look also exactly similar to each other, though they will have little bit differences. That is the monozygotic twins from the same blastocyst. If some of the cell get dispersed and after that they get implanted, then this is known as monozygotic twin. So here in that very much same time, another very important thing is happening. What is happening? This trophoblast will start to develop some finger-like projection, and whenever those finger-like projection go, and it will actually help this whole blastocyst to get attached to the mother's wall or the uterine wall, that actually go invaginate inside the uterine lining until it reach the blood vessel. So whenever it get blood vessel, it will stop. So those finger-like projection which is formed actually from this trophoblast, they are known as the chorionic villi. So chorionic villi are formed from the trophoblast, and later this same chorionic villi will form the placenta. See what happened, half of the placenta actually. So placenta is both maternal and the fetal. That means this chorionic villi will form the fetal part of the placenta, and the endometrium will form the maternal part of the placenta. So you can see wherever the inner cell mass are present. So here suppose the inner cell mass are there, only from that region the implantation had taken place, and wherever the inner cell mass is present, that is known as the embryonic knob. This is known as the embryonic knob. So wherever this embryonic knob is present, only from that region implantation will take place. See, suppose here in this region this blastocyst will not get implanted. So wherever only the blastocyst have the inner cell mass, only in that region, from that region actually implantation had taken place. In that same time, this trophoblast, see, this trophoblast and the endometrium will form the placenta gradually, but for the formation of placenta, whole placenta formation will complete around 2 months later, and placenta will form the hormone progesterone. But here this structure get implanted after 7 days. In that very much same time, it required the hormone progesterone, and in the ovary, you should know, in the secretory or the luteal phase, it had the corpus luteum. So corpus luteum was secreting progesterone hormone, which will start to decrease. In the menstrual cycle, we have seen it. If suppose implantation had taken place, then it is not allowed that the corpus luteum will degenerate and will not release progesterone hormone. It is not allowed. So that's why this trophoblast will release a hormone that is known as the human chorionic gonadotropin. HCG hormone is released by, it will be released by trophoblast. It will be released by the trophoblast, which will actually give an indication to the corpus luteum, so that the corpus luteum do not get degenerated in the time period up to the formation of placenta. So when the placenta will be formed, in that period of time it will release the progesterone hormone. But for now, for till that period of time, this corpus luteum will be still present, and that is known as corpus luteum of pregnancy. It will continue the release of progesterone hormone till 2 months until the formation of the placenta. So that HCG hormone will be present in the blood also, urine also, and that can give the indication of pregnancy. So in the pregnancy test kits, what happens? The detection of pregnancy will be determined by the detection of HCG. So this pregnancy test kit actually determine whether that urine have the HCG or not. So that is known as the Gravidex test or simply pregnancy test. So in the pregnancy test or Gravidex test, you can remember this word, this is new to you. So Gravidex test or the pregnancy test actually indicate, or it will find whether HCG is present in the urine. In the blood also it will be present, so it will be determined by this HCG. So it's in the urine, and that will be by the pregnancy test kit or the Gravidex test. Now let's just see what is happening. First of all, we had the outermost cell layer, that is trophoblast. So trophoblast will be still present. They are the flat cells. So here it is present. So that trophoblast had the important function. What were the functions? First of all, it formed the chorionic villi. It helped in attachment. Here this is the chorionic villi, suppose, and here these are the maternal tissues. So maternal tissues are the endometrial tissues actually, which will help in implantation. So now just see here inside what we had, the inner cell mass. So these are the cells, inner cell mass. So that inner cell mass contain a number of cells. They are larger in size. And here in the same blastocyst we had a cavity also. And that cavity had the fluid, and that fluid actually had the nutritive fluid released by the endometrium. So this is the blastocyst that has been formed. After that, just see what is happening in that period of time. In that very much same time, what happened? Gradually it is happening. See, from that time period, suppose this is the trophoblast. What is happening next? So this trophoblast will be still present. Here, these are the trophoblastic cell layer. Now, what happened? These cell layers, they will actually undergo a division. See, first of all, these cells, inner cell mass, they will form a layer of columnar cells. They are known as the columnar cells. And just behind that, a layer of cuboidal cells will be formed. So here you can see, these are the columnar cells, and just behind that we had a layer of cuboidal cells. So what happens? First of all, from the inner cell mass, the cells will divide. They will form the columnar cells above, and just behind that they will form the cuboidal cells. Now in that same columnar and cuboidal cells, they are together known as the embryonic disc. So this embryonic disc is formed by both the columnar cell and the cuboidal cells. They got a typical term name itself. those columnar cells, these cells are known as the epiblast, and this cuboidal cells, they are known as the hypoblast. So epiblastic cell layer and hypoblastic cell layers, they are formed from the inner cell mass after some period of time. Now just see what happens next. In that next stage, see, this is A, suppose this is B, this will be C. What happens in the trophoblastic cell layer, that will be remain outside, that will be still present. Now this embryonic disc, they will at one period of time, they will slightly descend towards the dorsal line, towards the middle region, they will slightly go downward. So whenever those cells go downward, you can see here, then we can get to have a cavity here. So you can see that here there is a cavity has been formed, and this cavity is known as amniotic cavity. This is known as amniotic cavity, and in that same time the trophoblastic cell layer, see here, suppose these are the trophoblastic cell layer. So this trophoblastic cell layer will form a layer of amniogenic cell. So those cells, they are formed from the trophoblast, they are known as the amniogenic cell. So just see here, these are the amniogenic cells. This cavity has been formed by see, this cavity is known as amniotic cavity, which is having the roof formed by amniotic cell, and beneath it is going to have the epiblastic cell layer. One more thing I had to tell you, that those trophoblastic cell layer which is coming just above this inner cell mass, these cells, they are known as these cells. Suppose from here to here, these cells, they are known as the cells of Rauber, or we can also call these are the Rauber cell. So Rauber cells are the cells, they are the trophoblast itself present above the inner cell mass. So you can see here a cavity has been developed, amniotic cavity, and in that amniotic cavity the roof is formed by the amniogenic cell that is formed from the trophoblastic cell layer, and just beneath that the floor will be formed by the epiblastic cell layer. Gradually what happened, these cells, cuboidal cells, these are the hypoblastic cell layer. So those hypoblastic cell layer, they will go on dividing. They will divide, and after that it will, see what happened, they will completely spread, go on dividing and form a layer, once again a cavity. So this is a cavity. This cavity is known as the primary yolk sac. So this is known as the primary yolk sac. So in case of human, the yolk content is very much less, but then also this primary yolk sac will be formed after some time. Now just see what happened. We have this epiblast and the hypoblastic cell layer. Suppose here we will have two ends. So this end, this is the cephalic end. This is suppose cephalic end, and this is suppose caudal end. So what happened, from the caudal end, some of the cells, they will start to detach from the epiblastic cell layer. So just see what happens next. So let me draw here, this is A, this is B, this is C, and this is D. So after that what happens, see, this is the same trophoblastic cell layer will be still present. Which is forming now the placenta. Some of the cells formed from the trophoblastic cells, they are known as the amniogenic cells, which will form the roof of the amniotic cavity. So here we have seen the amniotic cavity is being formed. Here these are the amniotic cavity, amniogenic cells, because of what we will have a roof of the amniogenic cells. So these are the cells, epiblastic cell. So this form, these are actually the transitional cell, these are these are the columnar cell, and after that we have the cuboidal cells. So we have the epiblast and the hypoblast. Now this hypoblast, they will start to form a cavity here. This cavity was known as the primary yolk sac. Gradually it will start to decrease in size. So whenever it is going to decrease in size, this is known as the secondary yolk sac. Now just see what happens here. From this epiblastic cell layer, from the caudal end, that is here, this is cephalic end and this is the caudal end. From the caudal end, some of the cells start to detach, and here these are the cells. Suppose this get detached and it will form a layer. So what happens? It forms the extra embryonic membranes. We have two extra embryonic membranes here from these cells. They are known as this is actually the extra embryonic mesoderm. So this mesodermic cell layer, this will not at all form any of the tissues of the embryo, but it will help in the formation of extra embryonic membrane. See here, these are the extra embryonic mesoderm. So this extra embryonic mesoderm will form the layers that will be amnion and chorion. See, because of this region in between this hypoplastic cell layer and this extra embryonic membrane, we have a cavity. Also another cavity has been developed, and that cavity, this is known as the extra embryonic coelom. So a coelom is been formed. This extra embryonic coelom will start to gradually grow in size, and this secondary yolk sac, which is actually nothing but the same primary yolk sac, they will start to gradually decline in size. So this way we have the next stage. Let us see what is it. Let us see what is happening here in this next stage. Let me see. This is first of all outermost lining that is formed from the extra embryonic mesoderm, that is the chorion. This will be known as the chorion. The amniotic cavity, which will be lined by the amniogenic cells. So here it is forming another lining, extra embryonic membrane itself. This is the amnion, and inside it we will have the two cell layers. First of all, here the epiblastic cell layer, which are the columnar cells, they are already present. So here these are the epiblast, and just beneath that we have the hypoblastic cell layer. So hypoblastic cell layers, they are the cuboidal cells. So those cuboidal cells will actually form another cavity that will be the primary yolk sac, and gradually it will start to decrease. So whenever it is decreasing, then it will be known as the secondary yolk sac. So here this is the primary yolk sac, and this one here in between chorion and the primary yolk sac or secondary yolk sac, this is known as the extra embryonic. These are known as the extra embryonic coelom. So here these are the changes that will happen here in this stage. Now just see what happens next. Now I will draw only these cells. Forget about this chorion and amnion. Let's just see what are the things that is happening here. First of all, let me draw only those cells, epiblast and the hypoblast. See what happened. Those epiblastic cell layer, they will go exactly middle. That means they will go descends a little. So whenever they go descends a little, what happened? They will form a layer. They will form a primitive streak. So these are the epiblastic cell itself. So it will form a layer here. These are forming a layer of cells. This is known as the primitive streak. This is known as the primitive streak, and this primitive streak will decide the head and the anus of the baby. So here suppose the head will be determined, and here suppose the anus will be determined by this primitive streak. Now just see what happened here. From this primitive streak that is developed from the epiblast, some of the cells they will go invaginate. Some of the cell will invaginate from this side and this side. So here these cells, they are present, they are formed just behind the primitive streak. That will form a germ layer, that is ectoderm. So ectoderm is formed from the epiblastic cell layer itself. So epiblast from the primitive streak. Just behind that, some of the cell will go invaginate, and from both the sides, whenever it is invaginating, it will form the germinal layer, that is ectoderm. Already we have this mesoderm, that is endoderm formed from this hypoblastic cell layer, that is already present. So these are the cells of hypoblast that will go and forming the yolk sac, that will be also there. So these are the hypoblast. So this hypoblast form the endoderm. So endoderm and ectoderm is formed. After that, in between we will have a layer of cell. See what happened in this stage. These are the two germ layers are being formed, ectoderm and endoderm. Endoderm forms first and then ectoderm. So in case of triploblastic animals, we are the triploblastic animals. So in case of us, what happens? In between ecto and endoderm, some of the cells will be formed, and those cells, they are known as the mesoderm. So just see here, those cells they have been formed. This is known as the mesoderm. So above that we will have this amniotic cavity. That amniotic cavity will be still there. Just behind that we will have the secondary yolk sac, also it is present, and around that the chorion will be still present. So chorion will be still there. So this way all the three germ layers are been formed. First of all, from the hypoblast endoderm is been formed. From the epiblast ectoderm is been formed. Now in between ecto and endoderm, the third germinal layer in case of triploblastic animal, mesoderm is been formed. So this way all the three germ layers are formed. In this stage, the stage is known as gastrula. So gastrula means it's a stage when all the three germ layers they are been formed. First of all, what happened? From the epiblastic cell, they descends and form a layer of cells towards the dorsal surface. This is known as the primitive streak. So that primitive streak that will determine the head as well as anus, or as well as on the right side and left side what are the things that will be formed, that will be also decided by the primitive streak. Now just beneath that, some of the epiblastic cell layer invaginate and form the ectoderm. Already this hypoblastic cell layer forms, they are forming the endoderm, and in between ecto and endoderm we will have the third germinal layer, the mesoderm. And after that, from the ectoderm, endoderm and mesoderm, they will form their respective tissues. Suppose some of the cells, epiblast and hypoblast, they will decide later on, whenever it form the germinal layers, it will form their respective organs. From the mesoderm, the muscles, the blood vessels, blood connective tissue that will be formed. The endoderm, they will form the alimentary canal. The ectoderm, they will form the nervous tissue or the outer surface skin that will be formed from each of the germinal layers. What are the organs, what are the type of tissues they will be formed, you can see from different books. So that are the stages that will happen in the embryonic development, that is gastrulation. When all the three germinal layers are being formed, that is known as gastrulation. So here this embryonic development is completed. In this next video we will see the process of pregnancy. Then after that we will see parturition and lactation. Thank you.